All in business. Okay. In theory. All right. I believe we left off here last time. So we talked a little bit about what biological diversity was, or the contracted version, biodiversity. So in the narrowest sense, again, total number of species you find in a geographic location. But again, it includes so much more, and we're going to keep talking about that. We have a whole presentation on threats to global diversity, and I'll get into it a little bit more there. But let's just look at it from the straight species perspective, because we know that the only thing that you can really reach out and touch and grab is a biological species. Any other hierarchical classification level, from kingdom down to genus, you can't physically touch it. You can touch a representative of a species that is classified at that particular hierarchical rank, going from most inclusive to least inclusive, but the species is the only thing that's actually real. Subspecies, yeah, you can touch a subspecies too, because, it's that level. It's like species level and below, you can actually, reach out and touch it. But it doesn't do you any good as a biologist or a paleontologist or generally a systematist if there isn't a means to organize the things that we classify and name. So when you think about taxonomy and the actual business of naming species and organizing them into the existing classification scheme, if we don't have a system to actually make sense of it, it's like just throwing a bunch of books on the shelves in a library. And it's not really a library, because libraries have classification systems, so you can find things, based on a whole host of characteristics, for lack of a better way of putting it, for each book that's in that collection. So if we don't have a system, you really can't make sense of the biological diversity that's out there. So what we ended up doing, or not we, I should say, Carolus Linnaeus actually developed the classification system that we still use today. And he did this back in the 17th century, or kind of started in the 17th century, late 17th century, and then kind of hammered it out in the early 18th century. But the first systematic, or the first classification system that we basically came up with as scientists, courtesy of Linnaeus, was essentially, it was kind of like a three kingdom, or almost three domain-ish type system. We're currently using a domain system versus a 5 kingdom system, but I'm going to talk about both in passing today. But you've all probably played the game, 20 questions. And anybody not ever played the game 20 questions? Okay, good. So we used to play this game. We got these little computerized small versions for my kids when they were small, and then I would, give it to them, and then they'd play 20 questions in the car when we'd be driving out to this place or that place for a couple of hours, kept them occupied. It was great. But, you didn't need the little game to do it. So essentially, it's like one of the first questions: are you animal, vegetable, or mineral? That's like question number 1 for any 20 questions game. And in that kind of genre, for lack of a better term, regnum animale is, essentially kingdom Animalia. Regnum vegetabile is kingdom Plantae, and regnum lepidium is what we'd essentially consider rocks. So it kind of threw the geology side in there. So that was essentially the basic classification scheme. And then there were gradations within it. Because once you figured out, well, this is an animal, it's not a plant. Okay, we got all these different animals. How are we going to organize those animals together? But how do we keep them separate from other animals that aren't quite the same? So again, intuitively, if I asked you to organize your sock drawer, for those of you who have a sock drawer, I have a sock drawer. How would you do it? Sarah, how would you organize yourself? Well, you're in my line of sight, girl. Come on. How would you organize? Taylor's like, oh crap, he's gonna ask me something next. Yes, I will. Sarah, carry on.
Well, I'd probably take all the socks out, make sure they're paired up.
Okay.
And then, for me, I'd sort them by length because I have short socks.
Different socks for different things.
Socks.
Okay.
So
first category is sort by length.
Yeah. And then, warm socks versus not warm socks.
Okay. Another characteristic. Maybe color might be another one. So it's like once you get them into particular groups, it's like you're clustering them based on shared characteristics. And that's essentially how systematists do things. It's like we group like with like. And fundamentally, when you think about Linnaeus and how he classified, I mean, Linnaeus was probably he was the most prolific systematist that ever lived. He characterized, described, named, and classified over 10,000 species just himself. When you think about how prolific you can be as a scientist, I've essentially designated and classified 2 subspecies in 25 years because I've been busy. Dr. Felix, describe a species, so on and so forth. He's much more prolific than I am, but that's the thing. It's like to do 10,000 over the course of your lifetime, and again, it's a lot more complicated today to actually describe a new species or new subspecies because the criteria for delineating has become much more sophisticated. Even in the last 25 years since I started this nonsense, it's gotten significantly more difficult to really, provide enough evidence to say this is a distinct species from something else. Yet Linnaeus did it on the basis of phenotype. What does that mean? What's the phenotype? And again, this is like Gen Bio crap that I just want to probe to see who remembers what. So when I say Linnaeus used a lot of phenotypic characteristics, what does that mean? Physical expression of what? The underlying genetic traits. So you've got the genotypic characteristics of the genotype. So you're looking at particular alleles and combinations of alleles that equate to something that you see: eye color, hair color, digit length, tail length, whatever, something along those lines. But until relatively recently, you couldn't really look at the genetic characteristics of taxa and use that as a classification tool. That only started probably about 30 years ago. Well, how long, generally speaking, has genetics, modern genetics, really been a big field? I'm thinking back to a seminal day. Anybody ever hear of the Human Genome Project? If not, you've been under some lepidium. You've been under a rock. No. Okay, so everybody's heard of the Human Genome Project. When was the Human Genome Project completed? It's not really completed, but it's completed, that they've sequenced the entire human genome. Anybody remember when? The calendar year was 2003. It was published in Nature on April 8th, 2003. What event 50 years prior to that, published in Nature on April 8th, 1953, was significant to the field of genetics? There were 2 authors on that paper. There were thousands on the actual Human Genome Project paper. 2 guys. They're both dead. Well, no, one's still alive. Cold Spring Harbor Labs out on Long Island. Well, not the discovery of DNA, but what did they describe in that paper? Who were they? They're James Watson and Francis Crick. So Watson and Crick, they get a lot of credit for like the whole DNA thing, but fundamentally what they did was describe the three-dimensional structure, the helical structure of DNA, which explains how it can actually self-replicate. It was a very short paper. It was like three pages, 3 or 4 pages. It's ridiculous, right? Considering the human genome one, it had like all kinds of appendices online because of the actual sequence information. 3 billion base pairs, sequenced. So, but again, Linnaeus didn't have any of that nonsense. He couldn't sit there like you guys did in BIOL 117, where you took a plant or an animal, you amplified a particular, you did a DNA extraction or tissue excision, DNA extraction, PCR amplification, you sequenced it, sent it off to GenBank up in the sky, and did a match to figure out, what species of plant or animal is this? Linnaeus couldn't do that. We didn't have DNA barcoding back at that particular point in time. So everything he did was reflective. All those phenotypic characteristics that he used to describe and distinguish one species from another and group species on the basis of similar characteristics, and then place them into this hierarchical classification from kingdom down to species, was all done on the basis of phenotype. No reproductive isolation experiments, no DNA nonsense. Didn't exist back at that time. There were some, notes relative to reproduction, because when you look at the natural history of any particular species, you get into some like breeding, particularly with domesticated animals, you can kind of figure out, I can't make a mule using a male and a female mule, right? Mules are sterile. How do you make mules? You breed a horse and a donkey. You never know which one they use as the female. Could be a size thing too, right? But realistically, that's the only way you can do it. They have fundamentally different chromosome numbers. They're diploid numbers. The 2 chromosome pairings, I forget the numbers for horses and donkeys, but they're 2 off. So the offspring have an uneven number of chromosomes, and for various reasons, you can't breed mules with mules. So it's kind of like, this indicates that under natural conditions, they can't or won't breed. But you can make stuff breed. And we'll talk about that when we get to speciation. But anyway, we briefly already talked about this hierarchical classification system. So you're going from kingdom all the way down to species. And in between kingdom and phylum, you can have subkingdoms, you can have superphyla, you can have subphyla, infraphyla. You have different types of gradations in between each of these, and depending on how distinct you want to make your groupings, you can apply, based on characters, these different gradations. You usually don't see them unless you're looking at the original. I'm going to say the term now and I'm going to define it later. Unless you're looking at the type description, TYPE, like type. Unless you're looking at the original description of that particular species, the systematist is more than likely going to place that species. They're going to do the whole thing. The systematist, also being a taxonomist, is going to name it and is going to place it in the existing hierarchy somewhere in here. I mean, we designate in most cases new species, but that new species has to be part of a genus. That genus has to be part of an generally speaking, has to be part of an existing family, that family part of an order, that order part of a class, that class part of a phylum, so on and so forth. You go up the chain. Sometimes you have to designate new ones. maybe there are no other species that are similar enough to the one you're describing based on a combination of morphological and molecular characters that you have to designate a new genus. Or maybe that new genus, that new species within that genus is in a whole new family. You know, sometimes when you have only one representative of a given family, we refer to this as a monotypic family. There's only one representative species. Usually we don't see that with new ones as much as relics. So it's kind of like maybe you have like Limulus. Let me get Limulus for you. I couldn't get the lab on platter. You probably don't know this. It's actually more than so this is a horseshoe crab. Some of you have seen it before. Flyweight has 6 foot rule, but I'm moving. Still moving. So this is Limulus. You'll get to play with Limulus in the lab. Limulus is in the phylum Arthropoda. It's also the only representative of its class. It's actually the only representative, I think, of its subphylum, class, and family. You know, it's a relic species. Only one left. And I've got 2 of them here. Went out and killed them. Now, you find them dead on the beach. But anyway, that's Limulus. That would be an example of a monotypic, representative of a monotypic family. You know, it's left over, you know, from way back in historical time. But this is still the system we use. Even though we've moved away from the 5-kingdom Whitaker system, when I say Whitaker system, this was a system of taxonomy and classification established in 1969. WHITAKER, Whitaker system. And it basically was composed of 5 kingdoms. Kingdom Animalia, Kingdom Plantae, Kingdom Fungi. It's like always, you know, trying to remember. Animalia, Plantae, Fungi, Protista, and Monera, Monera. And the Monerans were the bacteria. So you had Kingdom Monera, which was way off to the side. Then you had Kingdom Protista, you know, a lot of single-celled eukaryotic organisms. Then we had the fungi, then we have the plants, then we have the animals. So that was the 5-kingdom system that was established in 1969. Before that, we still fundamentally were following officially the 2-kingdom system by Linnaeus. Either you're an animal or you're a plant, and rocks are rocks. You know, we don't consider them in the context of assigning names. You know, there's still a hierarchical classification system that geologists use. Different system, but it's still hierarchical, and you're grouping like with like. So, you know, even within that 5-kingdom system, so you can see these 5 little blue squares here, you know, those are the existing kingdoms up until probably about early 2000s. In the early 2000s, we started getting into much more RNA and to a certain extent DNA-based, like late 90s, mid to late 90s, early 2000s, molecular systematics became much more significant in classification and taxonomy. It opened up a whole new, kind of a whole new field or subfield of systematics, and it created a lot of chaos in the system because a lot of people in the scientific community were thinking, this is the end all be all. It's like, now we don't have to worry about any of this nonsense of convergent evolution. What's convergent evolution? What's convergence? What does that mean? Again, going back to like the end of Gen Bio. Anyone remember convergent evolution? I know sometimes it's like we're going by stuff so fast at the end of a course, and then you're just like, I've had it. ooh, it's a 7-week term, forget about it. You're not going to remember any of this crap. But that's why I bring it up now. I always rehash these things. So convergent evolution is a process where, for example, two organisms may phenotypically look the same, but it's not because of shared common ancestry. It's not because, they're evolutionarily closely related. When you think about moles, the typical moles that you see doing the push-ups around here, Scalopus aquaticus, is probably the most common mole that you'll find in the area. Out here, you might find hairy-tailed moles. So the eastern mole, Scalopus aquaticus, digs through the soil, swims like this through the soil, right? When you look at marsupial moles, so we're thinking about two different mammals, but we're talking about two different, essentially, subclasses of mammals. Marsupials, where do we find marsupials? Where? Generally speaking. Australia, and where else? How many marsupials do we have in North America? One. Which one is it? Possum, right? Virginia possum, Didelphis virginiana. You see them usually dead on the road, or sometimes like you pull into your driveway and they're like, they're really just creepy looking critters. But they don't get rabies. It's one of those neat things, with possums. But anyway, you find a lot of marsupials also in South America. Not as many, and we're going to talk about why you find so many in Australia and so few in South America when we actually get into zoogeography in a little bit. But anyway, I digress. If you look at the marsupial mole, who is far removed phylogenetically, not even close, moles are placental mammals. They're more like us than they are the marsupials. Because we're placental mammals as well, based on the way we actually do reproduction. But if you look at the morphology of marsupial moles and you compare them to placental moles, there are a lot of similar characteristics. Their hairs are short. They're bidirectional. You know how you pet a dog, or you're brushing a dog's hair? It goes in one direction. And if you go in the opposite direction, it gets all screwed up. Moles aren't like that. Their hairs flip back and forth because they have to go forward, they have to go reverse, they're moving in tunnels, they're moving through dirt. It's not good to have long, unidirectional laying hairs. Their shoulder musculature, their shoulder skeletal structure is very, very similar to what you see in placental moles, because there's only so many ways you can kind of dig through the dirt doing the breaststroke. When you have similar characteristics, generally speaking, phenotypic characteristics that are similar, even though you're looking at really, really distantly related taxa, usually those characteristics are produced. They're still the product of natural selection, but they're the product of convergent evolution because the selective pressures of digging through the soil basically have optimum phenotypes. And even unrelated taxa, if they're subjected to the same environmental selective pressures, are going to evolve by this process of natural selection similar characteristics. And this is a confounding factor if you're using phenotype, solely using phenotype to classify like with like. Because if you had a marsupial mole and you had a placental mole and you were Linnaeus, you would put them in the same class, the same family, the same order, maybe even the same genus. Maybe you put them in a different subgenus, because Linnaeus didn't know about convergent evolution. So again, we're going to get more into this when we dig a little bit deeper into the evolutionary biology stuff in this first, small segment. But again, when you think about this classification system, the only thing that we've added in the last 30 years is the hierarchical level of domain. So it's a 3-domain system: domain eukarya, domain archaea, and domain bacteria. Archaea and bacteria are 2 domains that include multiple kingdoms, like a total of like 22 kingdoms between the 2 of them of just bacteria. And then domain eukarya has probably about 10 kingdoms in it now. We've split up the protists into like 5 or 6 different kingdoms. But, you know, animals are still animals, plants are still plants, fungi are still fungi. You know, they're still separate kingdoms. It's just we've kind of gone a little bit overboard. But we'll talk about that like towards the end of the next couple of slides. But know this order, this top-down order. In your lab notebooks, you know, if you look at all the dead stuff in the jars, on microscope slides, you name it, in your lab notebook, on each page where you actually have your drawing of the specimen, and you have all, and we're going to talk about this in lab today, you know, all that natural history information about what you see. What are the physical characteristics that define it? What are the characteristics that you can see to identify it? What does it eat? Where does it live? Who eats it? All of that, it's going to be down there. But above it, probably on the top left corner of that page in your lab notebook, you're going to have this classification hierarchy. And you're not going to have to know them all. You know, at a minimum, it's going to be like, yeah, everybody will know the domain because that's like a no-brainer. Kingdom, fairly straightforward. Most of the stuff we talk about, it's kingdom Animalia. Once we get beyond the protists and the plants, and then we get into the phylum and class. You know, we're going to talk about representatives of this phylum today in class. We're going to talk about phylum Chordata, for example. So we're going to talk about 3 subphyla, and in particular, we're going to really pound subphylum Vertebrata because we are vertebrates and they're the cool ones, right? They're the good ones that everybody likes to end on. But, you know, you've got 7 different vertebrate classes, and you have different orders within those classes. We're going to talk about those. So in some cases, you're going to need to know for the practical exams and the lecture exams what order this particular organism belongs to, what family is it in. But I'm going to talk about that in class. So you'll kind of be able to figure it out. You're going to be like, okay, he talked about this entire family of Schistosomidae. And again, we're going to talk about this in class. You're going to see it in that video, Parasites Eating Us Alive. You don't have anything better to do, like watch a lot of these videos ahead of time and take notes because they're all testable. But it's a particular family of blood parasitic flukes. And the fact that we talk about that family means, okay, I've got to, when I have that specimen in my lab notebook representing that family, we'll talk about 3 different species and the parasitic infections they cause. They were a pain in Napoleon's ass when he was taking over Egypt. You know, one particular species. We'll talk about all of these things, but it's like you have to have that information in your lab notebook and you have to kind of have it in your head. But a lot of this other info, it's not core because you can look it up. You can always look up the classification of a particular organism. I know a lot of these taxonomic categorical names because I teach this class twice a year, and you know, the ones that I work with all the time, yeah, I kind of know them because you have to indicate that when you're talking about them in publications in the literature. But if you remember this hierarchical order, domain, kingdom, phylum, class, order, family, genus, species, and you can come up with a little acronyms to remember it. Forget about domain because I remembered this stuff before domains actually were designated, but King Philip came over for good seafood, or King Philip came over for good sex. You know, whatever enables you to remember this is the order, you know, and after like a week, you're going to know the order. You'll never forget it because you're going to write it down so many times that it's just going to stick in your head. But again, when we look at this hierarchical classification scheme, we're going from the most inclusive. So if we're talking about domain eukarya, it includes all eukaryotic organisms. You know, includes the 4 traditional kingdoms. And if you split up kingdom Protista into like, you know, 5 kingdoms or 6 kingdoms, you're looking at 10 kingdoms worth of taxa. And if we're conservative and we go with, okay, there are about 2 million species of plants and animals and bacteria and protists that have been systematically described, properly systematically described and placed within a hierarchical classification system. If we're talking just about domain eukarya, we're talking about everything except for bacteria. So it's the most inclusive category. But by default, if you're including la-di-da-di everybody, right, as the saying goes, you're kind of like, my grandfather uses that saying, doctor, and I'm like, okay, I'm old. We've established that fact. I'm old, I'm bald. I also like did something with my beard, but you can't see it because I got the mask on, right? So it's a secret. It's a surprise. But anyway, you know, when you think about that really, really big category that includes so many different taxonomic groups, the criteria to be part of that group, you know, maybe just like a handful of them. Whereas in order to be classified all the way down to a species level through all of these different hierarchical levels, the numbers of characteristics go up and up and up. You know, there are defined characteristics that all animals possess. There are defined characteristics that all chordates possess. More specifically, if you're a vertebrate, you've got characteristics that not all chordates possess. Then we get down to the next lower category. Okay, we're going to look at classes. Well, if we're looking at vertebrates, there are 7 different traditional classes that all animals that possess a backbone, you know, we'll just pick on that one characteristic. If you've got this nice vertebral column, you're in subphylum Vertebrata. And you're one of 7 types of vertebrates. You know, are you a jawless fish? Or are you a jawed vertebrate? That's another big distinction. Do I have jaws or do I not have jaws? There are 2 classes of, there's one superclass broken down into 2 classes of jawless fishes that are living. What do we call a group of organisms that are not alive anymore anywhere? Extinct. What do we call the living ones? Also starts with an E, X, extant, E-X-T-A-N-T. So extant taxa are taxa that are still alive as a group. Extinct taxa, they're gone everywhere. There's one more gradation that I'm going to throw in there now because we're on a roll. If you have representatives of a species that used to occur in a particular area, but now they no longer occur there, they are not extinct there. It's what we refer to in the field as extirpated, E-X-T-I-R-P-A-T-E-D. And the only reason I'm spelling it out is because I used to love spelling bees when I was a kid. No, because it's a pain in the ass to write shit on the board for the people that are online, because then I've got to switch cameras and do all this nonsense. So I'm just going to spell it out. And if you want the spelling on something, or you want me to repeat the spelling on something, just shout it out. That, you know, that's anybody who's in the class, whether you're remote or face-to-face, because I know I'm a northerner, I talk fast, and I got a mask on, so it messes things up. So if a segment, likely a population of a particular species, maybe that species is wide-ranging, you know, its geographic range is most of North America, but in cases like this, sometimes we break those species up into subspecies. We have sometimes arbitrary designations dividing this subspecies from this subspecies in this wide-ranging species. Maybe it's based on geography. Maybe, oh, east and west of the Mississippi River, we have different subspecies of this particular wide-ranging species because its dispersal abilities are pretty limited. It can't swim across the Mississippi River. It's going to drown. Maybe it can walk across one of the many bridges across the Mississippi River. I've watched coyotes and raccoons and various critters walk across the bridges. The 2 bridges leading to Cape Cod. That's how rabies got to Cape Cod, by raccoons and coyotes crossing over the Bourne and the Sagamore bridges. But if they don't use bridges and their dispersal abilities are limited, maybe that river is a barrier in between those two subspecies. But generally speaking, if different subspecies come into contact with each other, they're not different enough to not interbreed. So subspecies can interbreed with each other, and when you interbreed, what does that do for differences between the two population segments? Do they become more different if they interbreed, or do they lose some of those phenotypic characteristics that allow you to define this one is different from this? They lose them. Gene flow, you've heard about this term before, and I'm going to talk about it again as we get a little bit more into it, and you're going to talk about it in population genetic stuff. When you have individuals moving from one population to another and interbreeding and exchanging genes, generally speaking, true or false, this is a good thing. So is it good to have movement of individuals from one population to another, to another, to another? Increasing gene flow? From like a natural selection perspective? Yes, of course it is. Because remember, when we think about what's going on out there, what natural selection pressures are acting upon the population, not all individuals in a given population are going to have the best set of characteristics to survive, right? This is basic Darwinian selection. This is what we talked about back at the end of Gen Bio. So only the strong survive, which is like the stupidest thing. It's like, maybe that's like an A-team statement. I'm Mr. T, only the strong survive. Okay, most of you are like, who the hell is Mr. T? All right, it was like a long time ago. He's like 70 now. He pities the fool. But anyway, who doesn't believe in natural selection. But when you think about natural selection, it's like, if you took all of us and without going down that Title IX road, if you took all of us and you stripped us naked and you threw us all outside, maybe some of you hairy bastards would probably do better in the cold than the people that have less hair on their bodies. I'm dead if I go out there. But all kidding aside, when you think about, it's cold out, if you have more hairs per square millimeter, and maybe they're longer and they can trap air particles in them when you fluff them up and you put them down, increasing the insulatory effect, and you have shorter digits and shorter tails and shorter ears, if you're in really cold climates, you're going to be less subjected to frostbite. You're going to survive better than somebody who's got like, I've only got my summer clothes, I saw the White Walkers coming from Tennessee. It's like, they're coming over the wall. No. But that's the thing. It's kind of like, not everyone in a given population is going to have the set of characteristics that are best suited to deal with those environmental selective pressures. And this is how you see shifts in phenotype and you see shifts in genotype when you're looking at the population genetic structure of what's out there in any given population. So if we have situations where you have reductions in genotypic and phenotypic diversity, maybe a disease comes through, maybe a non-native species that's a predator gets introduced and it eats up all the ones that look like a particular phenotype because it can see them better against the background. Or it can eat up the ones who can't climb up into the higher levels of the vegetation because they weigh too much. I usually have this problem when I'm trying to climb trees now. I can only get so high before I'm like, I ain't getting out on that branch because it's going to break. I'm trying to cut that branch anyway. That's the only reason I climb up trees now, in most cases. But those who are of slighter build, maybe have longer tails for balance, longer digits for climbing, they can escape the predators and not get eaten up. Those become the majority in the population. The other ones, they become the minority. And then sometimes they disappear. If the entire population in a particular area disappears, that population is extirpated. It's not extinct because Extinction deals with either specific subspecies or species. So like a subspecies can become extinct if that subspecies, for example, only occurred on Whitetop Mountain. The ones on Mount Rogers, they're slightly different. Maybe they're a different subspecies because those populations have been isolated above 5,500 feet because their vegetation that they live in has been isolated since the last ice age. You know, anybody who has not been up to like one of the high altitude mountains in the Southern Appalachians? Okay, good. What does it look like up there when you get up above 5,500 feet? What's the dominant tree type that you find up there? Are they like pine tree types or are they like oak tree types? They're pine tree types because once you get above a certain altitude, it's the same as if you were going farther north. So it's kind of like when you look at the vegetation on Whitetop or Mount Rogers or Grandfather Mountain if you're from North Carolina or Roan Mountain, you get up above like 5,500 feet, you see a shift in the vegetation type. You have like overlaps for a while and then all of a sudden you're like, oh, I don't see any more deciduous trees up here. It's colder. The humidity's different. 20,000 years ago, basically at the end of the last ice age, all of this area looked like the top of Whitetop, Grandfather Mountain. It was all essentially like what you would normally see for those of you who've traveled up to northern New England or Michigan or Minnesota or up into the southern tier of Canada. It's boreal forest. So, you know, when we start talking about habitat, we start talking about natural history of organisms and historical patterns of dispersal, zoogeography, you know, the geographic distribution of animals, phytogeography, P-H-Y-T-O. Phytogeography is the study of the geographic distribution of what? Plants. So when we start looking at geographic distributions of plants and animals, because that's something that we always talk about in the context of every group that we're going to talk about, they don't all occur everywhere. And when we look at the historical patterns of where they occurred and their movements over time, you know, this kind of helps you understand why we see them where they are now, why we see marsupials mostly down in Australia. And we see like, you know, four families in South America, but the vast majority, all the other families, are down in Australia. One representative of one of the orders is in North America, Didelphimorpha. So when we think about where things are now, it's always in the context of where they used to be and what the selective pressures over time did to affect the biodiversity of that particular group. So again, it's like, this is why biodiversity, it's more than just the number of species you find in a particular area. So when we think about extirpation, it can be really, really bad. For example, if you had a distinct island population of a species, doesn't occur anywhere else. It's unique to that island. It's unique to Whitetop above a certain elevation. It's unique to Grandfather Mountain. And for some reason, maybe it's climate change. Those oak trees and various, you know, maples and various other things, they're moving up the mountain with increasing temperature. It's kind of like if you were on an island and sea level began to rise. You're kind of like, this isn't good. Because at some point in time, it's like the surface area gets so small, maybe it's too little to support the number of species that actually are on the island, and then it gets really cutthroat with intraspecific competition. And again, you know, I'm throwing terms out that you may or may not remember from 117, but when we think about competition, competition within a species is referred to as intra-specific competition. Inter-specific competition is competition among, between or among species. And there's another clarification. Between is between how many? 2. Between a rock and a hard place. Among, 3 or more. And again, communication in writing and orally, it's important because if you don't use the right terminology, it leads to confusion. And when you get confused, it gets harder to understand what we're talking about. So if a population of a wide-ranging species that happens to occur at these high elevations, and it's a disjunct population. There's another term. It's like Dr. A's just throwing the shit out there. You're gonna be like, think of all those note cards you gotta make now, for these terms. If you do that thing. Disjunct, D-I-S-J-U-N-C-T. If you think about where you find things, if you find one population that's like, it's in Nova Scotia. I got one subspecies of one of the mouse species that I study, the white-footed mouse. There's one subspecies that occurs in southern Nova Scotia, and there's no other representatives of that species until you get down to like northern Maine. Yeah, mid-Maine, Penobscot River. That's weird. Why the hell is it way up there and there's nobody else of that overall species? It's a specific subspecies isolated to southern Nova Scotia. If something goes wrong and that subspecies that occurs nowhere else dies out, it's extinct. So again, depending on the context, that subspecies doesn't occur anywhere else, it's extinct. If that subspecies occurred there and some other places and that one population died out, it's extirpated. So we're going to use these terms, and you're going to read about these terms. You're going to figure this out when you start doing the basic informational search for your endangered species topic. Because again, if a species is listed as an endangered species, a lot of research has been done to designate it as an endangered species, which means more research has to be done in order to figure out how to keep it from becoming an extinct species. So if you haven't picked your species yet, this was more of a problem last semester when it was only 7 weeks. You still have like 13 weeks to go for the semester, but don't put it off too far, because otherwise, one, you'd be like, oh, I really like this one, I want to do it. Somebody's already doing it. Too bad. First come, first serve. So look at the list, look what's up there. It might give you some ideas. You're like, oh, that's cool. Maybe I can look at another big cat. Maybe I can look at like ocelots or snow leopards or something else. You're like, I wanted to do a tiger, but I'm like, I'll just do something else in the genus Panthera. Okay, so I digressed a little bit, but Panthera pardus, our friend here, this is the leopard. It is one representative of the genus Panthera. There are a few different species, 7, give or take, of the big cats. Lions, tigers, leopards. They're all in the genus Panthera. All of those cats, all of the different species of the genus Panthera, and other species that are representative of other genera. So again, plural of genus is genera. Species can be singular or it can be plural. It's a pain in the ass. But all of these different representative genera, they're all in the big cat family. Well, actually, that's old. It's actually Pantheridae, unless they shifted back to Felidae. But in any case, they oscillate. But you have different families in the order Carnivora. So all of the big cats, they're in the family Felidae. What about dogs? Dogs, wolves, coyotes, foxes. They're all in the family Canidae. They're collectively canines. But you have different genera within the dog family. You have different species within the different genera that are in the family of dogs. So again, it's just kind of like expanding as we go up. How many different orders? There's like maybe 20 odd orders of mammals that are carnivores. Or there are 20 different orders within mammal, 26 within Eutheria. But there are maybe a dozen different families that are in the order Carnivora. So again, and you have suborders, infraorders, superorders. You have all of these distinctions in between. You don't have to worry about that for our class. But when you take other classes, potentially a mammalogy class or a herpetology class, it's like the ologies. Mammalogy studies what? I'm a mammologist. I study mammals. A herpetologist, what does a herpetologist study? Reptiles and amphibians. What does an ornithologist study? Birds. Paleontologists study, rocks. No, animals and plants that became rocks, right? An entomologist. Those of you who had Dr. Bova, he's an entomologist. What does an entomologist study? Insects. So, protozoologist. I mean, there's an ology for every field. We're all biologists. We all study life, but subcategories, because most of us are jack of all trades. Anybody who teaches this class is a jack of all trades, because you got to know a little bit about a lot of things in order to get you guys together. But it's like there are certain groups of organisms, like Dr. Felix, he's an ichthyologist. He's also a paleontologist. He's a paleoichthyologist as well. But he's a fish guy. So he really knows about fish. But he's taking courses in mammalogy and herpetology because, that's the training that we got. We got a much more broad-based training 30 years ago than you guys get nowadays. Because everybody and their uncle is a specialist. You see this really in the medical fields. It's like, well, I'm a forensic proctologist. Think about what that entails. Probing the rectal anal area of dead people? Come on, it's a joke, it's funny. Forensic proctology. Okay, so remember this hierarchical classification scheme. It's a title somewhere, right? All right, so we've got our classification scheme. We want to group like with like. We know about convergent evolution. We know it's a problem. It's like, Jalen, I thought your arm was in a sling. But the black bottom's kind of like, did you break your arm? And you're like, no, not yet. Season's just started. All right, so we already talked about this stuff. Binomial nomenclature literally means the nomenclature of two names. So this is another thing that Linnaeus kind of hammered out in the rules. 9:20, right? It's okay, we still got like 11 minutes. So when you're designating a new species, that species has to have a two-part name, binomial. The first part of the name, it's kind of like your last name. If you did last name first, Argyros, George C., one each from the Army. But anyway, it's like, okay, the genus, you're assigning it to a particular genus within a family, within an order, within a class. It's like you kind of, once you figured out the characteristics of that particular taxon that you're looking at, you're figuring out, okay, is this something that we've already seen in some form or fashion? Or is it something totally new? Most of the time, it's not totally new. Maybe it's a new species, maybe it's a new subspecies, but the genus is probably already there. So when we use dichotomous keys, like we're going to use in the lab today, keys work great to identify known species. But sometimes the key can only get you to the genus, because you're just like, it doesn't have these characteristics that define it as this species. It's got some of these, it's got, oh, this is totally new. Sometimes the key can help you figure out, holy crap, I actually might have an undescribed species at this point, or an aberrant form. But if you have enough of them, if you've taken a series of specimens, you've trapped, like me, old school mammologist, I kill stuff. I don't just take photographs. I don't just take hair or DNA samples, I take the whole bloody thing. And I do all that stuff too, but it's like, you know, I'll tell you stories about that later. But if you've got all of these characteristics and you're like, it's something in between this species and that species, you may be looking at a new species. And it really kind of sucks ass if you've only taken a DNA sample and a couple of photographs and you let the critter go. It's not so much the same for plants. It's kind of like, oh, you know, plant biologists are always taking, you know, pieces of plants because they're like, yeah, it's a plant, it's going to grow. If you figure out you've got something new and you don't have the specimen in hand to do the complete systematic description, because you really can't just describe it on the basis of DNA. You got to do the whole ball of wax, which means you got to go back and go catch it again, which sometimes if you were on an expedition to a foreign country and now that foreign country is going through civil war, you're just like, I ain't gonna be able to get back there or I'm gonna get dead. So, you know, and I've known people who have made this mistake. They didn't take voucher specimens and they figured out they had something new. They went back the next field season and the area had been clear cut in rainforest and whatever was there is gone at that point. So it's kind of, you know, I always like to err on the side of caution and, you know, bird in the hand's worth two in the bush, right? So I digress. When we get into Linnaean classification, we get into taxonomy, we get into, I'm going to give this thing a name because it's something new, and I'm going to place it within an existing hierarchy, what genus does it belong to? What species does it belong to, or what species am I going to designate this as? So this is a two-part name. Because again, the species is the only thing that's real. The genus is not real. You've got representative species that are classified within a particular genus. But I can't reach out and I can't grab the genus Sciurus, tree squirrels, and a few ground squirrels within the genus Sciurus. I can grab an eastern gray squirrel and just pick one of them off of the road, you know, or a fox squirrel, Sciurus niger, two different species that occur in this area. More terms. So if you have species that are occurring in real space, and if you have two or more species whose geographic ranges overlap, anybody remember what the term for overlapping geographic ranges is ecologically? Starts with an S. Sympatric. You know, we're going to talk about sympatric speciation, but you need to know what sympatric is. So sympatric, when we're talking about where species occur geographically, it means their geographic ranges are overlapping. So gray squirrels, fox squirrels, they're both in the genus Sciurus. They're two different species that are distinct species. They're reproductively isolated. They won't interbreed under natural conditions. We're going to get to that, like what is a species in a bit. But if they're occurring sympatrically and they're not interbreeding, so you don't have hybrids, so a hybrid, or an hybrid because it's an H, a hybrid species is the product of two distinct species that interbreed. Generally speaking, it only happens in really closely related species. Sometimes if they're so closely related, hybridization can actually lead to essentially the extinction of one of the species because they interbreed and then you lose that distinction. But they were probably relatively recent splits. So anyway, if you're sympatric, your geographic ranges are overlapping. If you're parapatric, P-A-R-A-P-A-T-R-I-C, your ranges are abutting. They're not really overlapping, they're just coming edge to edge. And then if you're allopatric, A-L-L-O-P-A-T-R-I-C, your ranges are not overlapping. And when we get into allopatry, and then a little bit later, allopatric speciation, you know, how do species arise? This is all going to make a lot more sense, especially when we get to the point of reproductive isolation. This is kind of like, if you can't interbreed, can't produce viable offspring with another species, it means, well, you are distinct species because there's some mechanisms or mechanisms, plural, that are keeping you from interbreeding, even though you're sympatric in distribution. So again, we'll get to that later. So every species has to be referred to along with the genus name. If I ask you on a practical exam or on a test or a quiz, which species am I talking about here, or which species occurs in this particular area, you can't just say carolinensis. What the hell does that mean? Yeah, that is the specific epithet portion of the name. E-P-I-T-H-E-T. It's an adjective. So you have the genus portion of the name, which is a noun. These are the rules of zoologic nomenclature. It's the way it works. So the genus is a noun. The specific epithet is generally an adjective. So if you're talking about tree squirrels in the genus Sciurus, the genus is Sciurus, we're going to talk about the eastern gray squirrel, Sciurus carolinensis, described initially from the Carolinas. It was North Carolina. White-tailed deer, Otocoiles virginiana, or virginianus. Got a lot of deer down here. Literally, you go sliding down a hillside, you run into them. Sometimes that's why not only this is the first place we described them, but it's like they're so abundant down here. It's like, oh, this is why we have white-tailed deer, and it's named after the state of Virginia or the territory of Virginia, you know, when colonists first came here. Anolis carolinensis, the green antelope. It's not a chameleon, it's a green antelope. These things are running all around the Carolinas and farther south. Green ones and now invasive brown ones. Same genus, different species. Anolis sagrei, S-A-G-R-E-I. But you've got Anolis carolinensis, you've got Sciurus carolinensis, and then you have Blarina carolinensis, the southern short-tailed shrew. If I just said on a quiz or an exam or a practical, it's carolinensis because you asked for the species, I'm like, I asked for the species, not the specific epithet. The species can only be a species when you include the genus and the species part of the name. Because in conjunction, genus and species together, you're talking about a distinct entity. Because if we just say carolinensis, are we talking about a squirrel, a lizard, or a shrew? You don't know. It's like E. coli. Well, if I say E. coli, what are you thinking of? Capital E, because the genus is always capitalized, they are always italicized. If they're not italicized, you underline individually Sciurus, space, underline carolinensis. That indicates it's a Latinized name. Genus and species are always italicized because they're Latinized names. It's like, that's how you know you're talking about a genus and species. And the genus is always capitalized. But we don't know who E. coli is. Am I talking about the bacteria, Escherichia coli? Or am I talking about Entamoeba coli, the protozoa? There are situations where you can put S. carolinensis or A. carolinensis. You can never do it at the beginning of a sentence. That violates the rules. But if you're only talking about a bunch of different species in the genus Sciurus in this paper that you're writing, as long as you write out Sciurus somewhere before you abbreviate it S. carolinensis, S. niger, so on and so forth, then it's okay to do that except at the beginning of the sentence. If you specify any other genus that starts with the letter S, after that, and you start talking about squirrels again, you got to spell out Sciurus carolinensis or Sciurus and then the species that you're talking about at the beginning of that paragraph. Otherwise, are we talking about squirrels or are we talking about something else? So again, the rules are the rules to make sure everything is okay. All right, so we're going to stop here. It's 9:20. I'll see you guys this afternoon in lab. Generally speaking in lab, don't wear nice clothes. Wear closed-toed shoes. It's wintertime, I know, but You know, when it gets warmer, you're going to be like, ooh, I can wear my Tevas or the other ones. I just got a pair for Christmas for my daughter. But in any case, it's like, pull your hair back if you don't want to getting into dead stuff. Otherwise, I'll see you guys this afternoon. If you've already got your lab notebooks, great. If you don't, make sure you have them before next week. All right, see you guys at 1:50.